Dr. Hertz-Picciotto trained at Berkeley all the way through and has been at the Mind Institute and has really brought a new thing to autism that used to be thought of as exclusively a genetic disorder, but she has taken an epidemiological approach and shown environmental factors are very important. Alongside the people who have been looking at epigenetics, she has encountered the resistance of the field and the challenges therewith, but she is really the leader in the field of looking at the environmental factors in autism. Thus, others are beginning to follow suit, including our lab, and we really, really appreciate your coming here to share with us the latest of your work. And without further ado, Irva.
Thank you very much, David. It's really a pleasure to be here, and it's been a really interesting couple of days learning about the whole program here and interacting with undergrads and the other speakers and so forth. So thank you very much. So this is a picture, a drawing by a young gentleman, a teenager with autism. And actually it sort of starts, this is the full drawing, and as you get closer, you can see that this is it's the tree of life, and if you look at the fruit of this tree, you might be able to make out that there are these little fetuses in each of them, connected to the branches, and that at the roots of this tree is the detritus of, modern living, so to speak, old tires and the like. So in a way, this kind of summarizes the interest that I've had through my career in terms of understanding what are the contributing factors from the environment to early development of the fetus and then the young child. So that's what I'll be talking about. And the kinds of outcomes I'll be focusing on today are what we call neurodevelopmental outcomes, and these include 3 different domains, and really there are many more domains you can start subdividing. But the basic ones are cognitive impairments, behavioral deviations, and social deficits. The types of environmental causes I'm going to be considering really are root causes, we could call them. They're the causes that actually occur prior to the onset of symptoms, as opposed to, and sometimes when I talk to families or affected individuals, they're interested in talking about the exacerbating factors and things that might, after someone's been diagnosed, might be able to ameliorate symptoms or not. So that's not what my focus is. It's really trying to look at root causes of the development of the conditions and the behaviors themselves. And the conceptualization of causation I'm going to talk about a little bit in detail, but really this is the kind of disorder, and I think most disorders, most health problems really are multifactorial. There are a wide range of types of influences that occur, and the pathways can be very complex. So what is autism? Autism was first identified in 1943 by a psychiatrist, Leo Kanner, who described 11 cases that had come to his clinical practice. He was at Johns Hopkins, and he saw these common elements of these patients that included an inability to form social reciprocal relationships, difficulties with communication, and a pattern of repetitive behaviors or very restricted kinds of interests, and a sort of insistence on a ritualistic sameness in their lives. The symptoms are defined to be present by 36 months of age, or at least that's been the belief, although there is evidence that sometimes it does develop later. And it's also been considered to be lifelong, but again, there's a little bit of fuzziness here. There do seem to be cases where people develop, and by the time they reach adulthood, or at some point in adulthood, you really would not have known that they had the symptoms at a younger age. So these are somewhat controversial, but That's kind of the accepted paradigm is what's written up there. The pervasive developmental disorders actually include 5 different subcategories. At least this was under the DSM-4, which has now been replaced with DSM-5. But the original was these 5, and the top 3 are what we now, under DSM-5, don't make the distinction between autistic disorder, Asperger's, and pervasive developmental disorder not otherwise specified. The other 2 are much more rare and not included as ASD, even though ASD being autism spectrum disorders, those 3, but they share many of the same symptoms. The diagnosis of autism is made with 2 main instruments. These are the accepted standard in the field in terms of research reliability. Autism Diagnostic Observation Schedule or Scale is a standardized interaction, a play interaction with the child in which the examiner uses certain kinds of presses that are designed to elicit a predictable response. And I'll actually play a little video clip of one of those. The Autism Diagnostic Inventory is a long interview with the parent, the primary caregiver, and it really follows the whole trajectory of the child's development, and it can take about 2 or 3 hours to administer. And these 2 together give you both the snapshot in time of the child in face-to-face contact, and then the other gives you that trajectory, but it's based on report. So this is a child about 3 years of age, and he's being examined. You're going to hear, hopefully the sound will project well. Basically, the examiner is setting up there's going to be this little birthday party, and that little doll there, she's going to say, oh, it's baby's birthday, let's light the candles, let's sing the song, and so forth. And you'll see how he responds. And remember, a 3-year-old by this time has probably been to a couple of birthday parties and usually participates. So let's see if we can get that to him.
Now let's light the candles. Ready? Let's light them. Oh, it's hot. Oh, hot. Ready? Let's sing. Happy birthday to you. Happy birthday to you.
And he's playing with a piece of Play-Doh, which is the imaginary cake.
Happy birthday to you. Okay, time for cake. Baby's so hungry. Baby's so hungry. Time for cake.
You see, he's not very interested in the cake, in the imagination.
Can you give baby some cake?
Play.
She's hungry. Here you go, baby. Here you go.
So he dropped a piece of the Play-Doh cake, and you can see he misses the point that it's not a real piece of cake.
Not real. Come on back.
So this is one of the...
Baby's so thirsty. She needs a drink. Caleb.
Okay, so this is the sort of non-responsiveness, and parents often talk about having had a child who was responsive and then actually regressed, seem to have lost the skills, and seem to have, they'll describe the child as having retreated into his own shell, and they can't get through. So what is it that goes awry? What is it that is happening? And what is really going on in terms of the underlying cause here? So for quite a long time, there was the theory that it was actually bad parenting, and there was even this phrase, the refrigerator moms or the icebox parents. That is quite clearly not the case, but there is a legacy of that thinking that I think has created that sort of schism between the biomedical community and the community of people who are affected and are advocates for children and adults. So there is a neurological basis, a neurobiologic basis, and that's quite clear now. It's clear from electrophysiology studies, MRI studies, and the like, and autopsy studies as well. Definitely genes play a tremendous role, actually, in autism, and there's been a period of time where, and some people still say this, that it's really basically all genetic. This is the most inheritable health condition or mental health condition that there is. There are about 5 to 6% of cases of autism that are related to known syndromes that often manifest with autistic symptoms. That would include Fragile X syndrome, tuberous sclerosis, neurofibromatosis, Angelman syndrome we heard about this morning, Prader-Willi, and others. Then there are the single gene mutations and single nucleotide polymorphisms and there are hundreds of them that seem to confer some increased risk. And then the most recent literature has really been picking up on copy number variants, where there are segments of DNA that might be repeated 2, 3, 5, 20, 100, 500 times excess material, DNA, of sometimes small segments, sometimes large segments, or deletions being totally missing from the chromosome. And those two seem to incur some increased risk. We published a paper actually showing that the total length of duplications was substantially higher in children with autism than children without. But also many other factors. So what you breathe, eat, drink, medicines that you might take, products you use in the home, and then products that you might use on your body and your hair and your skin. All of these aspects of the environment, and then other things that really have to do with the mother's health that become part of the environment for the intrauterine environment for the child as well. So genes, instead of thinking of it as genes versus environment, I really like to talk about the genes and the environment because, again, this is multifactorial. It's probably true that every case, or virtually every case, has some environmental factors and some genetic factors. The genetic factors alone don't do it, and similarly for the environmental factors. So these little, what we call the sufficient cause pies, represents a set of factors, and it might be so over here we have a single individual, and it might be that A, E, and F are genes that she inherited from her parents. B and C might be factors that operated very early, say around the time of conception. There might be some influences during organogenesis in terms of the brain, CNS development, and some of those might operate through epigenetic mechanisms. And the idea of the sufficient causes is that if you take one of them away, you no longer have a sufficient set. So really, you can take action to remove any one of these things, modify it, and then you no longer have a sufficient set, meaning that there's a certain amount of resilience. Every individual has some resilience. We have lots of insults to our systems, and we handle them, right? We handle some of them up to a certain point, and then at a certain point, maybe you surpass that threshold of what adaptability there is. But the idea of multiple causes applies both across the population. So that might be her set of factors, and then there are, maybe dozens or hundreds of other sufficient sets that might operate within the population. And then when does autism originate? Well, I just alluded to the fact that the pregnancy period is very critical. We've had some discussion about that. And a lot of the findings from brain autopsy studies, really, they don't show necessarily really specific lesions, although I think some of the newer MRI studies are starting to really hone in on definite areas. But they are widespread, and it does suggest that, again, a sort of early origin for some of the aberrations in brain development. Connectivity is very important. And the genes, in terms of genes that have been found to be related to autism risk, there's clusters of genes that relate to synaptogenesis, and that, of course, occurs prenatally. But there is brain development that happens postnatally as well, and I don't think we can preclude the possibility of some factors operating actually in the postnatal period as well. So I think it's probably prenatal and postnatal and preconception. So what do we know? What did we know in 2000? And this is about when I was first brought into the field of autism research. I used to focus more on the perinatal reproductive kinds of outcomes. But I was asked to join a group that was submitting an application to NIH to form one of these centers on children's environmental health. And basically, late 2000, I was doing the literature review. I said, I'm not sure I'm really the right person. I don't know anything about this. And they said, it's okay. Actually, nobody really knows much. And I was a little skeptical. But when I did my PubMed search and really looked hard, this is really all we knew as of 2000, which is that there were these twin studies and family recurrence, sibling recurrence of autism studies showing definitely there's a familial component to this, probably genetic. And then there were also, there was just the beginnings of some candidate gene studies. And then there had been an epidemic of rubella in the US in the 1970s. And there were actually 2 independent case series which showed extremely high levels of autistic symptoms in those children who had contracted congenital rubella. And then thalidomide, which the data was from the 60s, but the papers were published in the 90s, showing that this medication that was used in pregnant women and was found to actually cause, really gross absent limbs, phocomelia and other things in the children who had been exposed. It turned out that some of those children also had autistic symptoms. And then valproic acid as well. So, so this is kind of a timeline of the research over the last, really 60 years. And just honing in a little bit on the early half of the 60 years. These are some of the publications from the 40s and then into the 60s. Really still a bit of argument going on as to whether there was an organic basis for autism or not. And then moving into some of those early factors. And then one of the studies I didn't mention, so there was the rubella. There was actually one study also looking at some other viral infections. But then, moving more forward in time, and I can't even read it here, there started to be more studies looking at, first of all, looking at the prevalence, and some studies really showing that the prevalence was rising, and then some strong epidemiologic studies beginning to appear really after the year 2000 here. So, and I won't go through all of these, but these are some of the real burgeoning epidemiologic evidence that's been coming out. But just going through, I think easier to see a list here. Okay, genetics, clearly a big role. Some evidence now, and again, really only mostly in the last 8 years, in fact, environmental chemicals. And there are now 8 studies on air pollutants, and virtually all of them are finding associations with autism. And I'll talk about our study on that. Pesticides also have received some attention. There are maybe 3 or 4 studies now, and 3 that actually looked at the same class. Some of them looked at other classes of pesticides. But the organophosphates, and in particular chlorpyrifos, which actually were banned for household use and production of products that are used in residential purposes, those are still used in agriculture, but several studies do suggest that people who are exposed to organophosphates have a higher likelihood of having a child with autism with prenatal exposures. And then maternal and paternal age, and I put here that these are possible proxies because is it age, or is it, what is it about aging that seems to put the child at risk? So yeah, it could be epigenetic changes that happen more with age. There is some there's some literature on that, and the de novo mutations in the father have been associated as well. But also, aging could be the accumulation of toxins in the body. In the maternal organism, it could be more autoimmune types of conditions that increase in incidence as women age. So age, I think, it's quite clearly associated with the risks for autism, but it's not clear actually what that means in terms of where we could intervene. And then medications, I mentioned thalidomide and valproate. Those, again, have quite strong evidence. There's also growing evidence that vitamins and maybe certain nutritional deficiencies in pregnancy could be related to autism risk, and I will talk a little bit about that some more. A couple of studies now, a couple of good studies, have come out with evidence about short interpregnancy intervals. So this is the time between the birth of one child and the conception of the next child. And that subsequent pregnancy then is the one that's at risk. And really a pretty strong risk if it's within 12 months. But even interestingly, up to 24 months is still at higher risk than those that were 24 to 36 months. So it seems to have a sort of dose response. And the thinking, I think, of people who have been looking at that literature and doing those studies is that this might be actually related to the nutritional status of the mother, that their pregnancy takes a lot out of the maternal organism and takes a while to replenish. And then the congenital rubella, I mentioned other infections. And then all the ones on this second page here there's a little bit of evidence, maybe a few studies, maybe not the strongest studies. And in some cases, there's a very mixed set of results. So smoking is an interesting example, which there seem to be studies that show smoking protective. There seem to be studies that show smoking is actually harmful. There seem to be studies that show no effect whatsoever. A lot of the studies, however, it's maternal report about their smoking, and so the actual quality of the exposure data is a little bit questionable. But this is one that I'm sort of watching closely because one of in my title I talk about quandaries. To me, I think this is a little bit of a quandary. We have air pollution and we have smoking. Well, the constituents of cigarette smoke and the constituents of air pollution are largely the same chemicals. They're the byproducts of fossil fuel burning. So PAHs, particles, metals, other volatile organic compounds. And it doesn't really make sense to me that air pollution, where you would get a lower dose, would not have an effect, or so equivocal seeming from the literature, whereas air pollution, we're seeing what seems like it's a pretty consistent pattern. And many of the studies, not all, but many of these studies did a pretty good job of adjusting for all the socioeconomic sorts of factors that tend to go with people who are living closer to freeways, are often in lower SES and may have other contributing factors going on. So, and then fertility treatments, acute conditions in pregnancy, fever and infection. A number of studies came out of Denmark, and it looks like that may hold up. And then the modern technology I have on here, this is really more one in the area of hypotheses at this point. Hormones is also of great interest, although it has not actually been investigated very much at this stage. So now I'm going to turn to some of the results. I'll talk about 3 or 4 examples and maybe try to connect this to the larger theme of epigenetics. When I was invited, I told David and Mary that I'm really not the right person for an epigenetics meeting. And they said, no, we want you here to give public health context. So that's my role here. Okay, so childhood autism risks from genetics and the environment, the CHARGE study. And the map here is showing California with the arrow coming from Sacramento And that's where the Mind Institute is located. So we have a catchment area that's kind of, an hour and a half or 2-hour drive to our clinic at the Mind Institute. And the goal here was that we set back in 2000, having looked at how little there was that had been published at that point in time, was really to identify causes and contributing factors for autism, including environmental, genetic, and their interactions. It's a case-control design, and what that means is that we, unlike an experimental study where you start with a group of animals or people or plants or cells, and you expose them and you then see what happens. Case-control study starts after people have a disease or don't have a disease. In this case, we actually had 3 groups. So we began with children who have autism. Then we took another group of cases of children who have developmental delays but not autistic features. And then the third group are our true controls. They're general population controls. We frequency matched them on age and gender and geographic region, but we actually find them through the birth files. And so they really represent the whole population of births in California, in this case, in our catchment area. The eligibility criteria are 2 to 5 years of age, born in California, living with a biological parent who speaks English or Spanish. We bring them all into the clinic, and that's where we administer both that ADOS and ADIR, the ones I talked about earlier. We also administer other behavioral instruments. We obtain cognitive developmental scores, adaptive behaviors, hyperactivity, a number of other of those. And then we do a full medical examination and medical history for the child, but also the whole family. Then we administer a rather lengthy interview asking about all kinds of behaviors and exposures. During pregnancy, we have what we call our index period that starts 3 months before conception and goes to the end of 1 year. And so we ask intensively about lifestyle, demographics, occupational histories, reproductive histories, and so forth. And then we collect specimens, and we get medical records. So it's a big operation. So it's a nested case control sampling design. Just to summarize, we have population-based recruitment, meaning we're not starting, as many studies do, with just a limited clinical population that happens to come to one particular clinic or set of clinics. We use standardized instruments to confirm all of the diagnoses. And then we're linked to a bunch of laboratories that we work with, and I'll talk a little bit about some of our collaborations with the immunology lab. So the slogan is to the families, be in charge. Okay, maternal metabolic conditions is the first one I'm going to talk about. And so the goal here was to really see if there were differences between the mothers of children who had typical development and the mothers of children who developed autism or developmental delay, looking at pre-pregnancy obesity, diabetes, either gestational or type 2, or underlying hypertension. And the rationale for looking at these, this was an idea of one of my graduate students, and I said to her, well, what's the evidence, what's the rationale? And she had done the research and really convinced me, yes, this was worth looking at. These are a couple of pathways that she had proposed. One is what you generally associate with diabetes, insulin resistance, and increased blood glucose of the mother, resulting in, because of transfer to the fetus, stimulating the fetus to become hyperinsulinemic. And then that leading to this rapid growth. And it's well known that babies of mothers who have diabetes tend to be macrosomic. And so there's this stepped up energy metabolism. And then that results actually in then the fetal hypoxia and potentially iron deficiency as well, both of which could lead to damage in brain development. And then the other pathway here, and there's more, I mean, these are just two hypothetical ones, is more of the inflammatory pathway where interleukin-6, which actually crosses the placenta, is elevated under these metabolic conditions, leading the fetal immune cells in the brain to also produce perhaps a sort of cytokine storm in the brain. And theories around inflammation, neuroinflammation, potentially a kind of chronic subclinical state there have been published. There's a paper that came out in PNAS a number of years ago really putting together a number of different strands of evidence that this may be contributing to autism. So these are the results, and we had both the self-report from the mother, but we also went back and validated with the medical records, and by and large, the self-reports were pretty good, but so we relied on the medical records when we had them and supplemented with self-report. So the odds ratio is the odds of someone with that particular exposure developing ASD versus divided by the odds for developing typical development, and the odds exposed versus unexposed. So great, if these were all 1, that would say that the exposure doesn't increase or decrease the risk for ASD or for developmental delay. But all of these, as you can see, are elevated, and most of them are have pretty good precision, this confidence interval not including the null value of 1, except in this case here. The overall picture is that if they had any of these, there was about a 60% increased risk of ASD and more than a doubling of risk of developmental delay. We also, I can do that here. Yeah. We also looked among both the ASD and the non-ASD to see whether some of the cognitive domains were influenced. And in fact, receptive language and expressive language were poorer if the mother had had diabetes. So although diabetes was not quite significant in the risk for autism, it was associated with these other domains, as well as reduced socialization on the Vineland scale. So the literature actually does support that the intellectual, a few studies, not a really strong, robust kind of literature, but there have been clues that diabetes does seem to be associated with intellectual disability in the offspring. So this maybe adds to that literature. And then I pose a couple of issues here on this slide. One is, certainly you can ask questions about mechanisms, but you can also ask questions about what is it that's upstream of those metabolic conditions in the first place. And in the environmental world, there's a lot of interest in what we call obesogens or diabesogens. And bisphenol A acts as a proestrogenic compound. Phthalates are anti-androgenic, and we're going to hear a lot more about phthalates in a little bit. Much of this literature is cross-sectional studies, however. It really needs to start getting a little more methodologically sound. And another factor that has been related to metabolic conditions is an interesting body of work that has come actually out of my institution by Kimber Stanhope and Peter Havel, who have done a series of experiments in overweight people, looking at putting them on different diets. And high fructose corn syrup is actually a pretty bad actor out there. We don't metabolize it the way we metabolize, the sugars that we're more used to. I think there's some plausibility that this may have something to do with the epidemic, may contribute to the epidemic of obesity that we have. It's a tough one because this is, food prices went down a lot when they stopped using sugar and, the cane sugar and so forth, and started introducing high fructose corn syrup. But, I don't know. Certainly the time trends match up, so it's an interesting one. And then the other little piece of data here that I thought was kind of interesting from the perspective of timing we talked a lot about the prenatal environment, but the gestational diabetes actually is associated with changes in breast milk composition. So breast milk contains a lot of things that people didn't know about. I mean, this is, again, a really growing area. But one of the things that it contains is secretory IgA. And secretory IgA is really key for the baby. It's what the good bifidobacteria in the gut love. And it really helps those populations to compete with the pathogenic bacteria. So it's very, very beneficial to the infant who is suddenly out in the world encountering new things, including a lot of pathogenic bacteria. It also directly regulates inflammation. This is a very small study, but it's really kind of interesting that it may well be that the diabetic mother, her milk has some differences compared to the milk of a mother who does not have that condition, and maybe that has some impact on the fetus. Also, just as a little thought experiment, is the idea of these time trends. And what else is going up when autism is going up, just as a kind of thought experiment. And you think about ADHD, mental health disorders, asthma has been going up for the last couple of decades, obesity, diabetes, autoimmune conditions, and autism. So is there some common set of environmental factors that might provide some kind of unifying explanation? And also, is it possible that the endocrine disrupting chemicals, like plasticizers or the high fructose corn syrup, may be increasing all of these conditions that then are operating to influence brain development? So it might be an actual pathway, or it could be separate pathways to all of these kinds of conditions. Maternal nutrition, I want to talk about for a little bit. So we looked at the intake of prenatal vitamins and the timing of intake, and we also looked at these in combination with genes that are related to folic acid metabolism, and looked to see whether the mothers of children with ASD actually had different intake during the prenatal period. And what we found is that, and then a little background. So folic acid is required for DNA synthesis repair, and it's really key for methylation. The prenatal supplements have already been recommended, right? We recommend these because they protect against neural tube defects. And that took a long time to figure out, but we got it right. And the supplementation is what women are recommended to actually start their prenatal supplements 3 months before they are planning to get pregnant. And in fact, we've seen a decline in the neural tube defects since food was fortified in the supplementation campaigns. So these are the results showing by month, starting 3 months before and then throughout pregnancy. And you can see that by about the second month, virtually, more than 80% of both the case and control mothers are taking the supplements. So once they know they're pregnant, they're taking it. But back here, the months before and the first month of pregnancy, there was actually quite a difference, with the case mothers being less likely to have taken those supplements for an odds ratio of 0.6. Those who took it, 40% lower risk. We also found that when we looked at the amount of folic acid, not just the supplements, but from the diet as well, that there was, in fact, a dose-response trend here. And this was important because there was a lot of discussion about whether there was hypermethylation going on. A lot of people were concerned that we were fortifying too much. People were taking the supplements and eating the fortified grains. Maybe that's not so good. So we went in with the idea of a two-sided hypothesis, that maybe high or low might be a problem. But what we found is that really even over 1,000 micrograms per day, which is a pretty high dose, there was no evidence that was harmful at all. In fact, that seemed to be the strongest amount of protection. And then I mentioned the interpregnancy interval, and so that's kind of, convergent with this idea of nutrition being important. And then the really exciting thing, because our data was based on maternal self-report after they already know what their child's outcome is. So there's always that possibility that the women who have the typical developing child are more likely to report that they took their vitamins than the other women. Although usually that recall bias that we call in epidemiology, it usually goes the other way. The cases are usually more likely to report everything, and the controls are the ones who usually forget things. So it wasn't, we didn't think it was plausible, but you couldn't preclude that. However, the really exciting thing was that this study in Norway that came out just in 2013 was a national cohort. They enrolled, I think there were 85,000 pregnancies that were part of this analysis, and they had, I think, their rate of autism seemed to be a bit lower than in this country, but they clearly saw, and the women were asked while they were pregnant at about 18 weeks of pregnancy, whether they had taken the prenatal vitamin supplements, and when they started and when they stopped. And indeed, they saw that same critical time window of that preconception, very beginning of pregnancy, and they also saw a 40% reduction. So it was it was almost shockingly validating that it was exactly the same amount of protection. So it appears, and again, this is a very, very large study and well done. So this is looking like it is probably going to stand up. So this is how folate starts up here. This is the one-carbon metabolism pathway. Folic acid has to get converted to folate, and one of the key enzymes is MTHFR. So all of the boxes are different enzymes in these metabolic pathways. We did some genotyping for all of the ones that are in the green boxes, and I'm just going to draw your attention to MTHFR, responsible for this conversion to folate. And then the folate goes through the methionine transmethylation pathway. Again, this is a methyl donor, and the methyl groups then get taken off and used for DNA, proteins, neurotransmitters under the COMT gene over there. So here's what we did. We looked to see whether the impact of folic acid supplementation was different according to the genotype of in this case, it's of the child. We also looked at the mother. And for the COMT gene, these are the relatively efficient transmethylators, okay? And in that group, there was some reduction. It was about a 45% reduction in risk, but there was a really large reduction in those who were inefficient, who had the particular genotype for COMT that does not as efficiently transfer the methyl groups. So there's clearly a subset of the population that is benefiting a huge amount from that folic acid supplementation. And then this is sort of a similar thing looking at the MTHFR gene, where this is when both the mom and the child have the wild type, and so there's really almost nothing happening here. In fact, if anything, but it's not elevated. So this is a very wide confidence interval. But this is if the mom or the child has the variant, and this is when both of them have the variant. So here again, that folic acid supplementation reduces by 70% the risk for autism. We've got a 0.3 odds ratio. Okay, you've heard a lot about epigenetics, so really now here's the way I'm going to talk about it. I was already pointing out that this is the methyl donor. But why that really early time period in gestation? Why that very, very beginning, or just right around the time of conception? Well, it turns out that the mammalian zygote actually undergoes an extensive DNA demethylation, and it begins 4 to 8 hours after fertilization. So here you have the fertilization. Here's the ovum. Here's the fertilization happening. You've got the zygote. Within 4 hours of fertilization, the paternal DNA begins to demethylate. Not 100%. Some of the imprinted regions and some other regions are not demethylated, but a lot of the methyl groups are pulled off of the DNA. That stays in that way for through all of these cell divisions here. It takes a little bit longer for to get to the 2-cell stage when the maternal DNA begins to come off. And while this is quite rapid, this actually takes a little bit longer. So the thought is that one is active and the other is a more passive sort of demethylation going on. And then just at the blastocyst stage, just around the time of implantation here, we see de novo methylation again. So all of those methyl groups then are added back on. Really interesting process. And then you know, things go on. And in fact, for the rest of this slide, I'm going to actually move over to this version of it, where, so here's what we just saw, the female and male pronucleus and the demethylation happening here. And then by, I believe this is around 12 days, the late blastocyst stage, you then have the primordial germ cells forming in the fetus for the next generation, and then there's an active demethylation that happens right here as well. So, and then for the sperm, you have the remethylation happening with the production of sperm, and in the female offspring, then of course it stays in that arrested meiosis for most of her life until she reaches the reproductive years, and not most of her life, but the whole early period. So, okay, so that may well be where it's really critical to have a plentiful supply of methyl groups during that first week, those first few hours, and that first week and a half of life. Okay, turning now to air pollution exposures. So again, we were trying to determine whether during the gestational period the child, the mother, was living near the sources of air pollution. So we mapped the homes where they lived. We collected these residential histories, and then we mapped those homes, and we mapped them and compared them to 2 different metrics of exposure. First one here is distance to freeway, traffic-related air pollution. We also looked at highways. And then the second was using a model that the U.S. Environmental Protection Agency has developed, looking at roadways, looking at other sources, regional pollution from manufacturing sources, and estimating, coming up with these isopleths of exposures to coarse particles, fine particles of 2.5 microns or smaller, oxides of nitrogen and ozone, based on those models. So again, we've adjusted for these factors: child sex and ethnicity, parental education, maternal age, and smoking. And what we find is that those who are living the farthest 50% being the referent group, that the odds ratios were really pretty close to 1 until you got within 0.2 of a mile, about 300 meters away from a freeway. And here we've got close to a 2-fold increase in risk. We also looked at the actual, as I mentioned, the regional air pollution measures, and looking at the fine particles here just as an example. For all pregnancy, again, about a 2-fold higher risk with, I think this is an interquartile range of the exposure going from the 25th percentile to the 75th percentile. And then similarly, actually, for the first year of life. So it didn't appear to be exclusively the prenatal exposures in this case. We also looked at modeled concentrations. So that was the second slide, was the modeled concentration. The first was the residential proximity. since, well, actually, one of these studies came out before ours, a couple of them came out before ours, and then several have come out since then, and there's another one that I think has just come out that I forgot to put on this slide. But there's now quite a few studies showing this association. The only one that didn't was the one by Kalkbrenner, but she didn't actually have typically developing controls. She was comparing developmentally delayed, she was comparing the ASD group to the developmentally delayed group. So since we've seen a lot of things where both developmental delay and ASD are affected by the same factors, that might be why that was a null study. And again, many of these studies controlled very well for socioeconomic status. So, and then the issue of why wasn't there a dose response? Well, interestingly, there is actually several exposure studies that indicate that the particle concentrations are high near the freeways, but they drop to background at about 300 meters out. And so that was our I mean, we actually didn't know that at the time that we made the cut point. We were just taking that you know, the closest, whatever it was, 10%, and just doing it within our distribution. But that explains why those middle categories didn't even show a slight elevation. I mean, it's consistent with that. You know, proximity traffic has also been associated with asthma. This, unlike the prenatal vitamins, was based on an objective measure. In other words, we weren't asking people to recall how polluted was your neighborhood. And PM 2.5, there is some evidence that it's associated with hypomethylation of placental DNA. And then we know certainly PAHs can induce all kinds of inflammation, endocrine disruption, oxidative stress. So these things are, you know, pretty nasty. Benzoate-pyrene, one of the primary PAHs in air pollution, has been shown in rodents to induce neurodevelopmental deficits. And then the very last example I'll talk about here is just a different kind of factor, maternal antibodies to fetal brain proteins. And this was in collaboration with Dr. Judy Vanderwater, an immunologist who was interested in whether the mothers of children with ASD, whether there was evidence that their immune systems reacted to fetal tissues. And they looked at many different types and found that fetal brain proteins were of great interest. We have 3 papers out now. And what we're seeing is that there's about 6 of these proteins that are you know, produced by the fetal brain. They're not produced by adult brain, and they're not produced by any other fetal tissues. And there are, it seems like there's up to about 23% of mothers of children with autism who produce specific patterns of these antibodies. So they do show up in in mothers of typically developing children. But if you look carefully at the data, there are certain patterns of them that seem to be fairly specific. In this particular set of patterns, it was fewer than 2%. It was about 1.3% of mothers of the children with typical development, as compared to 23% of the mothers of children with autism. So I think this is some of the other people have actually looked at some of these proteins as well. It's been replicated by actually a group at Hopkins, and fits with this idea of a neuroinflammatory kind of reactivity. The direction in relation to autism is unclear. Maybe it's because the child had something going wrong with their brains that they're, you know, producing more of, that these proteins are actually getting more into the maternal circulation. Or it could be part of the neuropathology itself that is producing the autism. So we really don't know the directionality here, but it's an area now of research. There is a possibility that some of this could be influenced by other environmental factors and by epigenetic kinds of mechanisms as well. Okay, so that's kind of just a few of the findings that we've had, and some of the thinking about what it means for mechanisms, what it might mean for interventions, and what it might mean for susceptible subgroups. We've been focusing on modifiable factors because our goal is really to intervene. And, I hope that, I will get to see the day when the rates stop climbing and they really plateau and start to really fall. And so that's what this is about. The limitation of the CHARGE study, I mentioned this retrospective data collection to complement the CHARGE study, the case control design. We have the MARBLE study, which is a longitudinal prospective study that takes advantage of the fact that there is this high recurrence rates in families. And so we enroll mothers who have a child with autism as they're getting pregnant. Sometimes we actually get them pre-pregnant. We follow their pregnancies and we follow the new child. We collect tons of specimens, and this provides really the complement to case control. It eliminates the problem of the differential reporting because the mother, yes, she knows she has a child with autism, but everybody in the study does. She doesn't know what the outcome of this next pregnancy is going to be, and there is this 18-20% chance that the child does develop autism. It also permits us to really examine timing of exposures with much more precision because of the way we're collecting specimens and the information we have. So I think that's it for what we have to say. It's just a brief thing. Things can go wrong in pregnancy as well as postnatally. And then, of course, all of this was a result of many, many collaborators and many, many staff people, and 1,700, actually, I think we're up to 1,800 families in CHARGE and about 300 in the MARBLE study. And this is a big burden to have us coming to their homes all the time and collecting this, that, and the other, but they are very dedicated participants. And thanks to the funders. And thank you.
To take a few questions before we move on to the next talk. David?
Just real quick, the antibody study, I was going to ask, when they did the monkey study and did the injection of the antibody maternally, and they induced the offspring repetitive behavior that Judy and Dave did, that kind of sold me on the causality notion. Is there still some equivocation left on that?
Well, I think we were pretty stunned by it. So what David is talking about was the monkey study. So the CHARGE study is actually part of a larger center, and there's been a number of other projects. One of them was a primate model. And so these were pregnant rhesus monkeys, and they were injected with purified IgG from, it was actually a pooled sample from mothers who had a child with autism. And I think it was just one, maybe there were a couple of injections, but they injected during the pregnancy, and then they have this huge primate colony at Davis, and they wean the rhesus babies at, I forget what age, but they're in these play groups, and they have kind of a surrogate mom and their own mom sometimes that they are united with for a certain number of hours per week. So after weaning, there's a break, and they don't see their mom for a while. And then they brought them back into this big cage, and they've got one of the surrogate moms on one side and then their real mom on the other side. And the pups that had been exposed to the purified IgG of human mothers with a child with autism started, it was just an amazing thing to see this videotape. So actually, first I'll tell you what the controls did. They kind of circled around one and a half times. They found their mom, and they went and huddled next to their mom and just sat there, and whatever, in the cage. Then we brought in the ones that had been exposed, and they just were circling back and forth and back and forth and back and forth 17 times before they finally settled and sat down with their own mom. And then there was another experiment where they brought them in with a playmate, and they've got a little bit of equipment to swing on, this, that, and the other. But the ones that had been injected with the purified IgG just went for this, like these little swinging bars and started doing backflips over and over and over and over. I mean, if that wasn't repetitive behaviors of no, purposeful kind of thing. I mean, it was, and to the point where the playmate was, it sort of reminded me of when I was on the playground and there was a kid who might have had some real behavioral serious problems, and we'd all kind of back away, and that's what this little monkey was doing, was kind of backing away, looking very suspect at the one that had been injected. So yeah, so that was the experimental model, and you didn't have to see the p-value to know that there was a really significant difference going on.
There's a question
over here. So in terms of the work you've done with the methyl diets, I want to discuss that a bit further because there's been a lot of interest in these methyl supplemented diets, like the folic acid, as you mentioned. I mean, I think we definitely know that they prevent notochord defects and other CNS defects. But there's also the possibility that they have indiscriminate action. I mean, there's no way to target, to get these methyl site and your particular gene of interest. It's going to go wherever it wants. It's a, random event. And in that aspect, so we've done a review on this, and we've also done some work in our animal with this. And we do cite your work, but there's also been the Rochester Epidemiologic Project where they looked at the same thing in terms of like how much folic acid the mom took and found the exact opposite findings, that if the more folic acid, the higher the risk of the autism. And in that aspect, then there was another study where they looked at these, I'm sure you may be aware of that study that came out last year of the Gambian woman, where they went in and they measured, they looked at the correlation between the folic acid they consumed. Again, this is in Gambia, we say give grain of salt, but then the metastable epialleles, like how many mutations. And they could find that the more likely that they were to intake folic acid, the more epimutations, DNA-methylate changes they had.
I didn't see that one.
And
so, yeah.
If I may just, and then our own research with the methyl diet in the rodent model is we can find greater repetitive behaviors, more anxiety. So again,
with more folic acid.
Mm-hmm.
So we just actually looked at some data on. We've just in the MARBLE study. We just started analyzing some of those data, and so when we just looked at what we were measuring, we didn't see a protective effect. I can't remember if we saw actual increased risk, but we did not. We saw it was very clear did not appear to be a protective effect. But what we did was when we then went back and we looked at their wait, let me get this right. Hold on a second. So I'm sorry, I'm a little confused here. But basically, what happened was when we looked at their genetic, their MTHFR genotype, then we saw that the ones who had, so the reason why, so actually what happened was there was not a correlation between what they said they took and what the actual folic acid was that we were measuring, the folate rather, that we were measuring. It was also true for some of the other B vitamins. There was not a relationship. But once we adjusted for their MTHFR genotype, then it all fell into place. So I think the real question is, maybe there seems to be a group that does benefit, and maybe, for whatever reason, we didn't see, it wasn't balanced out by the ones who didn't benefit and maybe that has to do with, I don't know what the distribution of the genotype of MTHFR might be in other populations. So that might be where you really have to do that. And if you haven't controlled for that, then you don't really know, what's actually going on, because the actual requirement seems to be higher in that group.
So it sounds like you're promoting a genes times environment equation, essentially.
Yes, a gene-environment interaction, exactly. Yeah, that was what I was trying to, I guess I didn't get that across very well.
So we'll hear Shauna first, and then there's one woman in the back, and then we'll go on with your talk.
Great talk, and I have one question, which you didn't actually mention anywhere, I think, the sexual dimorphism of ASD. Is this on?
No,
Thank you. So depending on the study, I guess it's 4, 5, 6 times as much,
about 4
more
times
prevalent
boys.
in boys than girls. So one of the things that I'd be interested in is knowing of all these risk factors that you looked at, air pollution, interpregnancy interval, blah, 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 which ones, if any, had a differential effect in boys and girls?
Yeah, most people get about a 4 or 5 or 6-fold ratio. Somehow in our study we ended up with, I don't know, it's like 7-fold. So we don't have as many girls as we would like to have. We actually did report the sexual dimorphism for our SSRI paper, the selective serotonin reuptake inhibitors. And there's actually 3 papers on SSRIs, but and we saw most of the effects seem to be in. I'm getting so old I can't remember anything anymore. I'm pretty sure we saw most of the effect in the boys, but in the girls it actually did, even though it wasn't statistically significant in the other direction, it looked like it was going in the other direction. But you can check that paper. But it is key. It is key. The hard part is getting large enough samples of the girls to actually be able to look at the heterogeneity there. But yeah, it is. And there's actually a lot of interest. There's actually, we contributed some of our data to Lauren Weiss at Stanford is doing a sex-specific allele study where she took our data and 6 or 7 or 8 or maybe 10 other studies to get enough girls to actually be able to look at that question.
All right, over one more question.
Where is the question?
You had global sensory disintegration on the list. We have a grandson who was diagnosed with it at 3. His mother worked intensively with him, got him all kinds of help. He's now a college freshman, getting all A's, has scads of friends, doing beautifully. Yeah. But where does that fall on the autism spectrum?
So it's one of the pervasive developmental disorders, but it's not considered part of the autism spectrum. So I had that slide where I showed the 5 different categories, and the top 3 that were on that slide, the autistic disorder, but they don't really use that terminology now anymore, but it was autistic disorder in the old DSM, Asperger's syndrome, and then PDD, not otherwise specified. So it's part of the pervasive developmental disorders, and with some similar symptoms as ASD, but it's not considered an ASD specifically. Yeah.
All right.
And congratulations that he's doing so well.
Thanks very much.